This method can help answer key questions about the cell therapy of the eye, such as what is the efficacy of a specifically designed bioengineered tissue in vivo?
With this technique, we provide a method for producing and culturing a retinal tissue, as well as a strategy for processing the tissue for eye implantation in an animal model.
Begin by washing 1 to 4 30 by 30 mm pieces of human amniotic membrane, or HAM, fixed in nylon scaffolds 2 times in 80 milliliters of PBS per wash in a 250 milliliter bottle on a shaker at high speed for 5 minutes. After the second wash, add 40 milliliters of working thermolysin solution to each bottle, and shake the bottles 2 times for 5 minutes at 450 RPM, followed by 30 seconds of vortexing. After the second round of thermolysin treatment, wash the pieces 2 more times with 80 milliliters of PBS per wash as demonstrated. Then, use long, sterile forceps to transfer the HAMs individually into a 10 cm culture dish containing 10 milliliters of PBS. Next, place one membrane with the nylon facing down in a second 10 cm culture dish of PBS, and detach 2 of the 4 clips holding the membrane to nylon. Insert the smaller ring of the culture insert between the nylon and the membrane, making sure that the membrane covers the smaller ring completely. Clip the second part of the culture insert to the top of the membrane so that the basement